So I want to thank the organizers for giving us the opportunity to present our work. So I'm going to be telling you about a new method we've recently developed called Primary Template-Directed Amplification, and how that method is enabling us to begin studying evolution at the cellular level. And so first, I just have a disclosure. Our new method, PTA, has been licensed to BioSkryb, which is commercializing the technology, and I am a co-founder, equity owner, and board member of BioSkryb. So I thought I'd start out by providing a little bit of context for our work. So as this conference is about finishing genomes, and as all of you know, over the last 20 or so years, we've been able to complete a number of genomes from many species as a result of the advancements in sequencing technologies. But those have all been done as a result of sequencing tens of thousands of cells mixed together in a bulk sample. So you have sufficient DNA for sequencing, or in the case of microbes, from sequencing a mixed population of cells or a pure population that was grown out in culture. But we know very little about the genetic diversity of the genomes within those populations. And so why should we care about that diversity? What is of interest with that diversity? And so, I think we should, what I've done really is just to step back and think a little bit about large multicellular organisms like ourselves, where we actually go from one cell into 30 trillion cells. And we know the mutation or copying error rate is about one error every 3 cell divisions. And so just during development, as we're developing into 30 trillion cells, there are about 10 trillion copying errors that occur. In addition, as you know, we continue to acquire mutations as a result of ongoing cell division in stem cells and other cell types, and as well as a result of exposure to things in our environment. In addition, those rates can be altered either by inheriting a germline variant or acquiring a somatic variant in genes that prevent or correct DNA damage. So, for example, people who inherit a p53 mutation have a higher background mutation rate. And so that's to say that within each of us, there's this enormous genetic diversity that occurs that we know, that we really haven't defined with measurements, and we know very little about how that genetic diversity contributes to human health and disease. What we do know is that at the most extreme end, one of those cells that acquires mutations can expand into billions of cells, and that's what occurs during cancer formation. And we know during that expansion process, there is also the acquisition of many, many mutations. And so in this now classic study of this kidney tumor, what they did was took bulk samples from different locations in the same tumor and did exome sequencing of, bulk samples mixed of tens of thousands at least of cells. And what they found was in each of those regions, there were distinct coding mutation profiles, suggesting even geographically, there's enormous genetic diversity. Interestingly, in this case, they each had mutations in SETD2, each of the locations, but they were all different mutations, suggesting that the tumor needed some sort of loss of SETD2 to be able to continue competing with the other tumor populations in different areas of the tumor. There's an analogous process going on within bacterial or other microbe populations. And so, when we do metagenomic sequencing, or we culture and sequence a population that grew from one cultured cell, we have a limited capacity to detect either cells that you can't culture in the first place or cells that are rare within the population. And so just to put this in context, most environmental bacteria are unculturable, and many of the 38 trillion bacteria in the human microbiome that each of us have living on and within us are also unculturable. And so that's really a large area of life that we have very little understanding of, and without really being able to isolate and sequence that diversity within populations, we really have a limited capacity to identify and study genes in these novel and rare bacteria, some of which could have value. to really understand intraspecies genetic diversity. And so, how does antibiotic resistance grow out, or how diverse or how common is it within a population of microbes? We have a limited capacity to understand how any two genes are interacting with each other in a given microbe. And then we also have a limited capacity to understand the kind of microbe interactions with their environment, with things such as the interaction of a specific phage with a specific microbe, and to connect the genome of each with each other. And so I hope I've convinced you that really studying life at the single cell level, or genomics, or the genome at the single cell level, is going to add enormous value and teach us a lot about genomics that are about evolution that we really have been unable to study up until this point. And so within the field, many, both in industry and academia, have been working on strategies for efficiently isolating and manipulating single cells. And we've kind of used this analogy of a microscope, from going from kind of bulk or gross level to the microscopic level. And we really need these tools that will allow us to get down to the cellular level. And they really have two components. And so one is isolating the cells, and there have been a number of microfluidic strategies, and there are new microfluidic strategies being developed all the time. And then the second is amplifying the genome of those, or transcriptome of those individual cells. And really the bottleneck for the field of single cell genome sequencing has been the poor quality of amplicons from the amplification reactions. And so that again is just making that point here, where you see kind of the gold standard multiple displacement amplification, where you end up having regions. So this is walking across the E. coli genome. You see regions where you have large overrepresentation, and then regions that aren't covered much at all. And so this uneven amplification with MDA has been a real challenge. And so in general, and I'll go into a little bit more background on all the genome amplification technologies, but in general, they all suffer from poor genome amplification uniformity and coverage, a lack of reproducibility, leading to people to select a subset of cells that look good. This all leads to increased cost. And then there hasn't been, like for single-cell RNA-seq, really capacity to do whole genome sequencing, or even amplify the whole genome in a massively parallel fashion. And MDA, the main issue with MDA is that it produces these very large fragments. And so if you add a barcode on the end, you really don't have a way to connect that barcode with all the downstream sequences, unless you do long read sequencing, which has its own challenges as well. And so just to give you a quick background on the field of whole genome amplification. So the first methods developed were really pure PCR-based methods, where you have either random or degenerate primers, and when you get two primers that sit down across from each other, you get product. And as I'll show you, this recovers a small portion of the genome, but in an even manner. The next group of methods developed were the isothermal methods. So really MDA is really, I would say, is the gold standard of whole genome amplification right now. And how it works is you have a random primer sit down, and then you have extension. You use this phage Phi29 polymerase, which has extraordinarily high processivity, a really low error rate, and really strong strand displacement activity. And as I'll show you in a couple slides, you get this branching structure, and then you get repriming and amplification, and you end up getting a jackpotting effect where wherever you prime first is amplified more than regions that were primed later. Subsequently, there were these hybrid methods, what we call hybrid methods, developed that use a little bit of isothermal amplification to add a common sequence, and then that common sequence is used for PCR priming. And so it's a little bit of isothermal, a little bit of PCR. And so again, these methods have had trade-offs that have limited their use. And so the PCR-based methods, so again, they don't cover a lot of the genome, but they're fairly uniform. but the error rate of the polymerase is much higher than the 529 polymerase, so they have a really high copying rate or false positive rate. The isothermal methods cover most of the genome, but in a very non-uniform manner, and then the hybrid methods perform kind of in between. And so I'll just discuss, briefly for completeness. Lianti was a method published a few years ago where they use a transposon to introduce an in vitro transcription promoter. They then amplified the DNA using in vitro transcription, and then they turn the RNA back into DNA and make sequencing libraries. And so this method hasn't really been widely adopted, mostly because the protocol is pretty complex. You have a lack of reproducibility, and so you really need to screen cells. You can't add cell barcodes, and then it has a really high false negative SMB calling rate, as I'll show you in a few slides. And so the approach we came up with was pretty simple, but quite powerful. And so we wanted to take advantage of all the great parts of MDA, which is the random priming, the extension, the strand displacement that allows you to get most of the genome and at high fidelity. But we wanted to limit this kind of jackpotting effect where wherever you prime first, you end up getting overamplification. And so the idea was pretty simple, although it took quite a bit of work to work it out and get things to work efficiently. But the idea is essentially you introduce an irreversible terminator into the reaction, and that creates smaller amplicons, and then those get displaced and they undergo less further copying because where they are primed can more easily become double-stranded. And so once they're double-stranded, there's no heating up, so there's no further amplification of the product. And so in theory, that would result in smaller amplicons that cover the more even manner. In addition, because you're not recopying the primary, the daughter amplicons as much, you should get less error propagation. And so in theory, and I'll show you some data that shows that that's actually what we see. So I want to thank the organizers for giving us the opportunity to present our work. And so we thought we had a great idea, and we had some initial experiments that showed a lot of promise, but we really wanted to know how good our method was. And so the first thing we did was really to do a bake-off, and over the last couple of years, we've done extensive comparisons to all the other methods out there. And so again, to review, we compared it to the pure PCR methods, different flavors of MDA, the mixed methods, as well as Lianti. And the initial approach we did was to perform scMDA, which is kind of a newer version of MDA and PTA on 10 cells. We used 1,000 genome cell line NA12878 because it has a good gold standard. And then we did the genome amplification and high depth whole genome sequencing on all cells, so we didn't pre-select cells. In addition, the Lianti paper had been published around that time, which had done a similar study on Lianti, Malvac, Picoplex, MDA, and DOP PCR. And so they also used a diploid fibroblast cell line. And so we downloaded their data and compared MDA and PTA in our hands to their methods, and then calculated various quality and variant calling metrics, again, using the bulk sample from each of these as kind of the gold standard for the associated single cells. So first we looked at percentage of reads mapping, and so you can see again we see consistently PTA has 97 to 99% of reads mapping, and so it's not producing a lot of junk DNA. When we look at the mapping quality, again you see for PTA is similar to MDA, not quite as high as the bulk sample, but again producing higher quality amplicons, especially when compared to the methods that use more PCR. But when we really got excited was when we got this result, which was looking at increasing number of reads and the fraction of the genome covered. These are the two bulk samples. We can see that PTA approaches the same amount of coverage with the same amount of sequencing as a bulk sample. And these are actually 10 cells here. The error bars are quite small. But what you can see is that we're really approaching getting the same depth and quality of sequencing from a single cell that we get from tens of thousands of cells mixed together. And so what this looks like across the genome, so this is SEMDA, and this is walking across chromosome 1. This is the centromere that doesn't get amplified very well. But again, you see these big spikes and valleys, so areas of high coverage and areas of low coverage. And so again, when you go to sequence, you sequence a lot of this and very little of the areas that are covered, not so much. With PTA, again, with the same bins walking across chromosome 1, again, we see that the centromere isn't covered so well, but we see much more even amplification, and we don't see those drops to 0 that we see with MDA, and so much more even amplification. And so when we quantitate that using a Gini index, which is a measure of uniformity, again, we see PTA has a lower Gini index. So these are the two bulk samples. So the lower the Gini index, the more uniform. And again, we can see that PTA has reproducibly, you see very small variance here, and consistently more uniform than all the other methods. But what we really care about is our ability to detect variants accurately within single cells. And so what this is showing you here is when compared to the corresponding bulk sample, so these are the two bulk samples, we ask what percentage of the variants detected here can be detected in each of the single cells or the subsampled files. And so what we see is PTA is able to call 80 to 90% of single nucleotide variants detected in the bulk sample, which is in comparison to 50 to 60% in the next best sample. And I will say we can also call these with very high precision, so in the range of 99% plus precision. The reason that we're able to do this is that PTA is amplifying both of the alleles much more evenly. And so when you look again at the heterozygous sites, in the bulk sample, you see this Gaussian distribution around an allele frequency of 50%, as you would expect. And you can see PTA, when compared to all the other methods, is both not losing as many alleles, but also maintaining that allelic balance much better than any of the other methods that are available. And that's really, again, another measure of our ability to be able to amplify the genome in a much more even manner. And then one other interesting and important feature of PTA is it allows smaller templates to compete with larger templates. So in this case, the mitochondrial genome with the same amount of sequencing is being covered about 400x with PTA, which is significantly higher than the other methods are able to cover the mitochondrial genome. And with just low-pass sequencing, we can actually do copy number calling and get really accurate mitochondrial genome sequencing. And so there are a lot more details that I don't have time to go into, but our paper's been posted on bioRxives, and I would encourage you to go there to really kind of dig into the details of lots of advantages that PTA has over existing genome amplification methods. But I was just going to spend the last couple minutes just touching briefly on some applications we have. So now that we have this better capacity to study and sequence and really get close to finishing the genomes of single cells, we can begin to go back and ask questions that were previously inaccessible. Such as we know these tumors are heterogeneous, but how heterogeneous are they? And so to get at this question, we had been working with CD30/40 positive cord blood cells, where we take one cell, let it expand, and then isolate individual cells that should be identical. And then when we find variants that are seen in multiple cells, we know that they were acquired somatically, but not seen in the bulk. We know they were acquired somatically. Whereas if it's only in one cell, we know it's likely a false positive. And so with these 6 kindred cells, you can see we get a consistent estimate of about 0.33 somatic SNVs per megabase, which translates to about 1,000 SNVs in these blood stem cells from the time of fertilization to when they become cord blood cells when the babies are born. And so we've used this approach to begin to measure the genetic diversity in single cells in cancer samples. And again, the story has been really, when you look at bulk studies like this bulk cancer sequencing study published a few years ago, that things like leukemia and some other childhood cancers have really low coding mutation rates per megabase. But again, this is all done at the bulk level, whereas adult cancers have significantly higher levels. But what we've done is gone back and done bulk sequencing, and then from those same samples, isolated single cells and done single cell whole genome sequencing and estimated the actual number of variants per single cell. And so in the bulk sample, because you're averaging out all the cells, you're losing a lot of the variants that are limited to a subset of cells. And as a result, what you're ending up with is a much lower estimate of the true genetic diversity. And so what we see actually is we measure in these leukemia samples somewhere around 0.2 coding mutate, or these are actually whole genome sequencing, so mutations per megabase. But we actually see about 10-fold higher mutations per single cell, and then you would multiply that by the total number of cells within that population. And so what you can see is there's actually vast, genetic diversity that's not being captured by these bulk samples. In addition to measuring genetic diversity, we can also think about measuring mutagenicity. And so we've, again, used in this case hematopoietic stem cells and exposed them to a mutagen, in this case the alkylating agent ENU. And you can see at increasing doses of ENU, we see increasing number of mutations per cell. But we can then also go on to look at the locations and the context of those mutations. And in this case, we've identified some new patterns of ENU-mediated mutagenesis. Further, we can begin to think about trying to prevent genetic diseases. And so this has been a collaboration with Barry Baer. But the idea is, again, not a new idea. So pre-implantation genetic testing exists, where you isolate cells from an embryo. But again, the difference here is that we have much more accurate sequencing as a result of our amplification of those small number of cells that are isolated. And we can, in this case, you can see multiple biopsies, and then the embryo is the gold standard and consistently call copy number change. But we've gone on to do whole genome sequencing and can actually call variants across the genome in an embryo prior to implantation. And then finally, we've kind of looked at genome editing. And so again, I think there's a lot of value to looking at genome editing at the cellular level as tissues may be limited and your amount of editing may be different between cells. And so you really want to measure the per cell off-target rate. And so we've used that with a couple different guide RNAs. One which is known to be more promiscuous, one which is less, and you can see all these translocations induced in different cell types in this VEGF guide RNA versus only one really that we saw on this EMX1 guide RNA. We can do analogous, we have analogous strategies for looking at indels and other types of genetic variation. And then just to circle back to microbes, we've begun using PTA and C, again, looking in the environment, but also looking within human samples. We can see consistently that we are able to get much more complete genome assemblies compared to MDA. And so this is in collaboration with Ilana Brito at Cornell, where she's been working with MDA for some time, and you can see we get much more complete genomes using PTA. And then finally, just looking forward a little bit, and beginning to think about that mutations occur within a cellular context. And so what if we can measure both the cellular context and the genotype within the same cells? And so Again, using PTA to amplify the genome and existing methods to amplify the transcriptome. And within the same cell, get a sense of the cell state as well as the mutations that occur. And they've done some work at BioSkryb with the FLT3 inhibitor quizartinib, where they can look at the grow out resistant cell lines that will grow out over time, and then compare the parental cell line to the resistant cell line and see gene expression changes, as well as, in this case, the acquisition of a specific FLT3 mutation that's mediating that resistance. And so more to come on that soon, but I think the combination of DNA and RNA is going to be extremely valuable. And so in conclusion, I hope I've convinced you that single-cell DNA sequencing is really poised to transform our understanding of cellular evolution in cancer biology. I think for many applications, people will switch over to single-cell DNA sequencing. I think eventually microbiome studies will be done using single-cell sequencing. We believe that PTA has really overcome the bottleneck of the field. And believe that outside of cancer and bacterial sequencing, there are a number of different applications where PTA can be used. And so with that, I'd just like to thank all my collaborators and funders. I am currently recruiting postdocs. If you're interested, send me an email. And I would encourage you to take a look at our paper just posted on bioRxiv for more details. And I thank you for your attention, and I'm happy to take questions.